Hello guys, welcome to my YouTube channel Raise Science Simple, and let's make the science simple. Um, Currently I am giving lectures on the population genetics, and I have already mentioned about the random genetic drift and linkage disequilibrium in my previous videos. So if you have not watched these videos, please go and watch these videos before watching this natural selection video. And without doing any delay, let's start the topic of natural selection, which is also known as the Darwinian selection. If you see this term, the natural selection, what do you understand by this? The natural selection, as its names imply, the selection that is done by the nature. So nature is selecting the most fit individual. If the individual is most fit, it will survive more, and if it will survive more, then only it will reproduce, and if it will reproduce, it will produce the offsprings, and if it will produce the offspring, that will take part in the evolution process. So natural selection also take part in the evolution process. And how the nature is selecting that the particular genotype is more fit or less fit, that is on the basis of genotypes. If the genotype is more fit, or if the presence of a particular genotype is advantageous for the survival of the organism, then the nature is going to select that genotype. So with these notes, we can start. The natural selection term was used by Darwin in the book. The name of the book was The Origin of Species, and that was written in year 1859. Now let's see the definition of natural selection. As you can see, natural selection is based on the idea that the organisms that are more fit or that are best suited to their environment, they are more likely to survive and reproduce, and this process causes species to change and diverge over time. And how the natural selection occurs? There are three rules. Number one rule, there will be more production of the offspring in comparison to the survival and the reproduction. Then organisms differ in their ability to survive and reproduce on the basis of their genotype. Number three rule, in every generation, genotype that promote survival are going to be selected by the nature, and then they will take part in the process of reproduction, and in this way they will produce the offspring, and that will help in the evolution process. So in every generation, genotypes that promote survival in the current environment are present in excess at the reproductive age, and thus contribute disproportionately to the offspring of the next generation. The progressive genetic improvement in population, because if there is an evolution, there is definitely improvement in the genetic material of the population, and that will result from the natural selection, because nature will select the genotype which is more fit, and it will constitute the process of evolutionary adaptation, or it will participate in the process of evolution. So if you see this whole definition, the progressive genetic improvement in populations that results from the natural selection take part in the process of evolution. So there are two kind of generation. One is the discrete generation, and another one is the continuous generation. In the discrete generation, the organism reproduce and it die, but it produce the offsprings, and but these offspring can never meet their parent. Why? Because parent will die just after the reproduction. That is the unlucky part, and this is known as the non-overlapping generation, or you can say the discrete or non-overlapping generation. Okay, then the another one is the continuous generation, in which the organism will reproduce and it will produce the offsprings, and these offsprings can meet their parent. So there is an overlapping pattern. So this is the overlapping generation. Okay. Now how the nature is selecting the best fit individual on the basis of its genotype. However, the process of natural selection in the haploid and in the diploid organism that differ. In the haploid organism, there is only one set of the chromosome. In the diploid organism, there is two set of the chromosome. One set of the chromosome you are getting from one mother, and the another set of chromosome you are getting from father. And again, in the haploid organism, where there is only one set of the chromosome, there are two kind of generation. One is the discrete or the non-overlapping generation, and another one is the continuous generation. In both of these generation, the rate of change of the change in the frequency of the alleles are going to be different. For example, in case of the discrete generation, if you see, this is the bacterial cell type A, this is bacterial cell type B, and if you want to know the number of bacterial cells A and B after T generation, you can use this formula where AT and BT is the number of A cells or the B cells, bacterial cells B after T generation. Omega is the relative fitness, A0 is the number of bacterial cell type A at 0 generation, and B0 is the number of bacterial cell type B at 0 generation. So how the allele frequencies are going to change, and if suppose you know the number of bacterial cells after T generation and number of bacterial cells after 0 generation, you can calculate the relative fitness of a particular allele to the another allele, or the particular genotype to the another genotype in case of the haploid organisms. So how there is going to be the change in genotype frequency? It's not proper to say the bacterial cells. We will mostly quote in terms of the allele frequency. So the variable of interest is usually the allele frequency and not the population size. So therefore, that P and Q denotes the frequencies of genotype A and B respectively in T generation. Then you can calculate the frequency of you can calculate the frequency of genotypes A and B after T generation by using this formula, where you can see what is PT and QT. PT and QT are the frequencies of genotype A and B at T generation. PT is the frequency of genotype A, QT is the frequency of genotype B after T generation, P0 is the frequency of genotype A after 0 generation, and Q0 is the frequency of genotype B at 0 generation, and omega is the relative fitness, and T is the number of generation. So basically, in this particular example, the relative fitness is very important, which is omega. You can decide which genotype is most fit to the particular environment in comparison to the other genotype, and that's how the nature is selecting this. This is the mathematical model basically. And the another term is the selection coefficient, which is inversely proportional to the omega. If the particular organism or the particular genotype is most fit, the selection cannot go against that particular genotype. So selection coefficient is the measure of the relative strength of the selection acting against a genotype. So a selection coefficient will be low if omega is going to be high. With this note, you can read this whole problem. I'm not going to read this whole problem. What the basis of this whole problem is this, that there are two kind of strains which contain two types of genotype. One is the RM77C and another one is the RM43A. And these genotypes are coding for a particular enzyme which is the phosphogluconate dehydrogenase, and which take part in the metabolism of gluconate but not in the metabolism of ribose. And you want to see which allele is more fit in comparison to other fit when the bacterial cells you are growing in the presence of gluconate media or in the presence of ribose media. To be simplify this, these are the two alleles, 43A and 77C. Both are coding for the 6PGD. And you want to see if the bacterial cell you are growing in the, if suppose this is the bacterial cell and you are growing in the gluconate media, and when you are growing the same the bacterial cell in the ribose media, then which genotype is more fit in comparison to the another genotype. Okay, so for this, what we can do, you know, the frequency of RM43A they have given. At 0 generation, and similarly this is the RM43A at 35 generation, and this is in the presence of ribose, this is in the presence of gluconate. Okay, when you know the P0, which is 0.455, you can calculate the Q0 for the gluconate media, okay, which is equal to 1 minus 0.455. Okay. Similarly, you can calculate, because PT is given, which is 0.898, then you can calculate the QT will be 1 minus 0.898. What is QT here? This is the frequency. Q is the frequency of RM77C. Okay. P is the frequency of RM43A. Okay. Okay. Now, you know the PT and QT, you know the T, which is 35, and you want to calculate the omega, which is the relative fitness, and you know the formula, which is like log PT by QT is equal to log P0 by Q0 plus T into log W. So you can simply write the values of PT, which you know is 0.898. This is QT is 1 minus 0.898. Then P0 you know is 0.455. Q0 you know is 1 minus 0.455 plus T is 35 into log W, and log omega, if you will do the calculation, you will get the value of omega, which is 1.0696 in the gluconate media. Similar thing you will do in the ribose media. And the ribose media, what happened? Let's do for the ribose media. This was in the gluconate, and then you do for the ribose media. And now you are comparing RM43A with the RM77C allele. So one bacterial cell will have the RM43A genotype, and the another bacterial cell will have the RM77 genotype. And you want to see which bacterial cell is more fit if you are growing the bacterial cell in the presence of gluconate media. That's what your purpose is. Which genotype is more fit? Then in the ribose media, you know the P0, which is 0.594. You can calculate the Q0, which is 1 minus 0.594. Similarly, you know the PT, which is 0.587. You can calculate the QT, which is going to be 1 minus 0.587. And if you will put these values here in this formula, you will get the log of 0.594 divided by 1 minus 0.594 is equal to log 0.587 divided by 1 minus 0.587 plus 35 log omega, and omega you will get like 0.999. So you can see that this particular genotype, or the bacterial cell that contain this particular genotype 43A, will be more fit because the relative fitness is more in the gluconate media. So the bacterial cell that contain this particular genotype will be more fit if you are growing that bacterial cell in the presence of gluconate media. However, in the case of ribose media, this particular genotype is not comparatively more fit than the other genotype. Okay, so basically you are comparing the two alleles that codes for the GND, which is the enzyme. This is the phosphogluconate dehydrogenase enzyme, and you want to see which allele is more fit in comparison to the another allele. Okay. So now coming to the continuous generation or the overlapping generation. So continuous generation, it means the parents will produce the offsprings and they will not die after just reproduction. So this is known as the continuous continuous generation. And in this you can calculate the M, which is the relative fitness with the continuous growth. Omega is the relative fitness with the discrete population. If you know the value of omega, you can calculate the relative fitness in the continuous generation by using this formula. Then coming to the diploid organisms, in which one set of the chromosome you are getting from mother and another set of chromosome you are getting from father. So let's see, omega W1, W2, W3, they represent the Darwinian fitness of the genotype. Capital A is for W1, capital A small a for the W2, and small a for the W3. Okay, and W1, which is the fitness of the genotype A, in that case W1 is equal to 1, W2 is equal to 1 minus HS, and W3 is equal to 1 minus S, where S is a selection coefficient and H is the degree of dominance. So here you can see the Darwinian fitness of the genotype. Capital A is independent of the selection coefficient. So what is the viability? It's a probability that the genotype survive from fertilization to the reproductive age. Means the zygote is formed and then the organism is not dying, it will grow till the reproductive age, and it will take part in the evolution process. Then the absolute fitness. If the fitness of the one genotype is independent of the another genotype, you are measuring the absolute fitness. Okay, for example, the genotype AA, the fitness of the genotype AA is independent of the fitness of the genotype small a. In that case, you are just measuring the absolute fitness of the each genotype. Relative fitness, when you are comparing this particular AA with capital A small a or small a, so the value of the viability of each genotype relative to the another genotype. So here you can compare this dominant genotype with the recessive genotype, dominant genotype with the heterozygous genotype, like that. This is the relatively how fit particular genotype is in comparison to the other genotype. So this is the survival rate. The overall survival rate is the percentage of individual born that survive to reproductive age. Because if you know there is a survival, it means they should survive after the reproduction, till the reproduction age or after the fertilization. Reproductive rate for any given genotype or phenotype, the average number of offspring born per individual. If you are the suppose an individual, how many offspring you have, that decide your reproductive rate. Relative fitness, when you is the survival and or reproductive rate of the genotype related to the maximum survival or the maximum reproductive rate of the other genotype in the population. So in this you can see this example. For example, survival is 10% for this genotype, 10% for the heterozygous phenotype, and 20% is for the recessive homozygous genotype. Here you can see the reproductive rate is similar. What does it mean? If the individual is containing the DD genotype, is producing only 4 offspring. If the individual is containing heterozygous genotype, then only it will produce the 4. The average number of the offspring is 4. And similarly, if the individual is containing small d, then the average number of the offspring that is going to be produced is 4. So reproductive rate is same here. So how will you calculate the relative fitness? You will calculate the relative fitness for this particular genotype, which is the survival rate, which is 10%, divided by the maximum survival rate, which is in the case of this recessive genotype, which is 20%. So 10 by 20 is the relative fitness of DD, capital D, capital T. Then again 10 by 20 is for heterozygous genotype, and 20 by 20 is for homozygous recessive genotype. So this is the relative fitness, and as you can see, relative fitness is high for this particular genotype because the survival rate is also high for this particular genotype. In this case, the reproductive rate was same. if you see this case, in this the survival rate is same. So what you are going to do, relative fitness, how will you calculate for this genotype? You take the number of reproductive rate for this particular genotype, and then you divide this number with the maximum number. If you see the 8, 8 and 8, the maximum number is 8 only, so you will divide 8 by 8. Here for this genotype 8 by 8, or for this genotype is 4 by 8. If suppose if I write this value, let's say here 10 instead of 8, then I will do what? I will do 10 by 10, 8 by 10 and 4 by 10. Okay, I will take the number that is the reproductive rate for this particular genotype, and then the maximum number. So the if you compare 10 and 8 and 4, you will see the 10 is higher value than in comparison to 8 and 4, and therefore we are dividing it by the maximum number. So that is the definition also says, is a survival and or reproductive rate related to the maximum survival. So maximum survival here is 20%, or the maximum reproductive rate here is 10. So like this, so we can calculate a relative fitness. What if both of these numbers are different? You need to multiply that. Like for example, if you multiply 0.1 into 10, it will come 1.8 and 1.2. If you see the maximum number is 1.2, so you will just do what? 1 divided by 1.2, 0.8 divided by 1.2, 1.2 by divided by 1.2, and that's how you can also calculate the relative fitness if the survival rate and the reproductive rates are given to you. So this is the change in allele frequency in the diploid population. Okay, so these formulas are very important. Here you can see change in allele frequency in the next generation is equal to p square omega 1 pq omega 2 divided by omega asterisk, and the value of omega asterisk is this one. Similarly, the frequency of the another genotype or the another allele, which is q prime, is going to be equal to pq omega 2 plus q2 omega 3 divided by omega slash. Okay, and omega 1, omega 2 and omega 3 we have already have seen there. Omega 1 is the relative fitness of capital A, omega 2 is the relative fitness of Darwinian fitness of capital A, small a, and omega 3 is for small a, small a. Okay. With this note, can you solve this equation? What does it mean? If you see this problem, assume a value of p is given. Number one thing, p is given, so p is 0.168. so definitely you can calculate q, which is equal to 1 minus 0.168, and if you calculate this value 1 minus 0.168, it will come, let me just do that, it will come like 1 minus 0.168, which is 0.832. Okay, they have also given the value of omega 2 and omega 3, and omega 1 is 0 because the CY allele is lethal. So you will get three kind of genotypes: C by CY, which is lethal, so they are not going to be produced. Then you will get C by plus, which is heterozygous, they will survive. And then there will be the plus and plus, that is also going to survive. Okay, now you can calculate the frequency of the CY allele. So p prime, if you will calculate, p square w1 plus pq, you know the pq 0.168 into 0.832, and omega 2, which is omega 2 is giving is 0.5, and then the omega asterisk that we need to calculate. Here you can see omega 1 value is 0, so this is 0, so we will get the p slash value is equal to 0.168 plus 0.832 into 0.5, and if you will calculate the omega asterisk value, on this basis. So how will you get this value omega solutions? Again, omega 1 is 0, so this term is going to be 0. This term is going to be 0 because omega 1 is 0. So we will calculate 2 into 0.168 into 0.832 into omega 2, which is giving 0.5 plus 0.832 square into omega 3, which is giving 1. And when you will calculate this value, you will get, let me calculate this value. It will come like 0.832. If you will do the whole calculation, you will get the values, which is 0.832. So here you will put the 0.832 value, and the value of P prime is going to come is 0.084. Now the question is, because CYCL allele is lethal, and they are asking what is the expected frequency of this heterozygous genotype. So this heterozygous genotype, the frequency of the CY allele is exactly the half of the plus allele, because this particular frequency of the CY allele is going to be 0. So only CY that is contributing to the survival is in the heterozygous genotype. So the frequency of P prime, if it is like 0.084, then the heterozygous genotype plus is also going to be the 0.084. So it is the plus CY, and plus, if you will do, it will be like 2 P prime, or you can say 2 into 0.084, and 2 into 0.084 is going to be 0.168 is the expected frequency of this particular heterozygous genotype. What will happen if we will say that this CY allele is not lethal? Then you will follow the exact protocol of the Hardy-Weinberg equilibrium, because in that case omega 1 is not going to be 0. So you need to calculate the frequency of CY plus, and then plus and plus. All of these are going to be survive. Okay, and in that case you cannot say that the frequency of the CY is exactly the half of the plus allele, because in that case this is also going to be survive. And for that you can calculate the allele frequency by 2 into CY, number of individuals that contain this, then CY and plus divided by 2n. This is the frequency of allele CY. If you want to calculate the frequency of allele plus, then also you can calculate in the same way, which is like 2 into plus CY plus divided by 2n, because in this case CY allele, if it is not lethal, it is going to survive. In that case the frequency of CY allele and the plus allele is going to be different. Okay, so that's how you can solve the equation. And the last thing is the marginal fitness. We have read about the absolute fitness, where the one genotype, the fitness of the one genotype is independent of the another genotype. Relative fitness, when we are measuring the fitness of a particular genotype in comparison to the another genotype. And then there is a marginal fitness that you can decide by the multiplying the probability of the allele being in the genotype. So let's say probability of the allele multiply by fitness of that genotype. That is the marginal fitness, how fit that genotype is and what is the probability that particular genotype is going to be there in the organism. If you multiply this, you will get the marginal fitness. Okay. So for example, if you know A alleles are found in capital A and capital A small a in the proportion P and Q, therefore the marginal fitness of capital A containing genotype is equal to the probability of that particular allele in the genotype into relative fitness of that genotype, which is omega 1. That's how you can calculate the marginal fitness for a particular kind of allele. Okay, and that's all for this lecture, and uh, we will discuss in our next lectures about the genes in the population genetics. So do watch my channel, and thank you so much. And if you like my content, please subscribe my channel, and thank you so much.